Ai ho mai kai mai na o
Okay, aloha. Welcome everybody to our monthly Slice of PI-CASC seminar series. We typically hold these on the first Tuesday of the month at noon here in Hawaii. And this is hosted by the Pacific Islands Climate Adaptation Science Center, or PI-CASC. I'm Brad Romine. I'm the Deputy Director of our PI-CASC University Consortium. This Slice of PI-CASC seminar series provides a platform for sharing ongoing and state-of-the-art climate adaptation research and science to management applications in Hawaii, the US affiliated Pacific Islands, and beyond. This presentation is being recorded. I'm sure you saw a notice of that if you're online, and will be available on the PI-CASC website under news and events, along with previous seminars in this series. For those joining us online, please keep your microphones on mute during the presentation. We will have time for questions following the presentation. Please feel free to leave those questions in the chat along the way if you're online with us, and we'll address them at the end. At the conclusion of our seminar, please do complete a short survey to help us in planning future PI-CASC seminars. The survey will pop up after you log off from Zoom. Before we get started, I want to thank Rachel Lenz for her work in organizing the seminar, along with Hal. And for her baking skills. I believe there is a pie in the back of the room, or will be. Okay, awesome. We're going with our Slice of PI-CASC theme here. I also want to thank Kristen and Lisa for arranging lunch for us. If you're in the room with us, haven't grabbed lunch, please go ahead and help yourself as we get started here. And I also want to thank Katie for making the Hiʻilei today. So mahalo for that. So it's my pleasure and a big mahalo to introduce our four speakers. Thank you for making the trip over from Heʻeia today. Wish we could have come over to your beautiful site today, but we really appreciate you coming here. First, I'd like to introduce Dr. Shimi Rii. Shimi is the research coordinator with Heʻeia National Estuarine Research Reserve, or NER, and also with the Hawaii Institute of Marine Biology. She's originally from Manana and Waipio, Oahu. I had to look up Manana. Is that Rabbit Island?
Pearl City.
Pearl City. Okay. I thought there was an inside joke there. Yeah. Thank you. And she has a research and education background in marine biology and oceanography. She also, little known facts, played violin in high school and ukulele in a few college luau's.
Because of the band theme.
Yeah, we're going to keep with the band theme here. And because of the pie theme, she noted she loves apples but hates apple pie. So we do have, again, a pecan pie today. We're also joined by Dr. Kaleonani Hurley. Thank you for being here, Kaleo. Kaleo is a postdoctoral research with Heʻeia NER. Oh, yes. Yes. Thanks, Katie. And the Hawaii Institute of Marine Biology. Kaleo is originally from Mililani, Oahu. And sticking with our musical theme, Kaleo played trumpet in the marching band in high school and sang in a band in college. And she's also known as the Crab Lady and Crab Whisperer. I'd be interesting to know a little bit more about what that's all about. Next, we have Hiʻilei Kawelo. Hiʻilei is the founder and executive director of PI-CASC Heʻeia. From Kahalu'u, Oahu. Is a student in the art and science of Lawaia, or fishing, and Kaneohe Bay her entire life. She has a background in zoology from UH Manoa. And she is also this year's Holo Holo No Bolo fantasy football commissioner, of which I understand Brenda is a part of as well. To segue to Brenda Asuncion.
Commissioner.
Oh, I see. Brenda is the Hui Malama Loko'ia coordinator at Kua Aina Ulu Aumo, or Kua. And she's from Waipio, Oahu. Education in marine biology, and back to our musical theme, because I understand was a percussionist in an high school band. So welcome you all. Oh, Brenda, yeah. Pass it over to you, Shimi. Get us started if that's how we're doing it. Yeah, thank you.
Ready? Where's Zoom? Okay. Does it work? All right. Thanks, Brad, for that introduction, and thank you guys for all being here today. So we're gathered here today to talk about a few projects. There's a lot of projects in Heʻeia, as you guys know, but today we're specifically talking about DNA. And you guys know it doesn't really stand for decoding natural assemblages, but we're trying to be cute. So, today we're going to talk about DNA as a tool, eDNA as a tool for research in Heʻeia. So we'll highlight some projects that looks at really who's there in Heʻeia, and then talking about how extreme events impact the community in Heʻeia, and then also looking at some food web dynamics, and then lastly, how we're working to increase community access to the DNA. So before I even get into those projects, I want to orient all of us to Heʻeia, this beautiful place and a special place that we all get the privilege to work. This picture was taken by Kili'i Kotubete, also at Paipai'o Heʻeia. And this place looks like this because of a community-driven action, because of all the kupuna and the people who care about this place allowed it to look like this right now, so that we can persevere and perpetuate Aina Momona, and then also community resilience. And before we even start, thinking about why we do this, it's really because of some of the kupuna that is shown here today, Auntie Alice, Uncle Jerry, Auntie Carolyn, and then also many of the organizations that work together, all of them together to preserve Heʻeia in the form that it's in. And we work to honor the knowledge, the wisdom, and the life experiences of these people. So in 2017, we were designated a National Estuarine Research Reserve. So that means it's a NOAA Office for Coastal Management, and we're part of this national system. So we're in the 29th, we're actually the 29th, and there's already 30. Connecticut became the 30th last year. And then, so each of these, as you can see, the black dots are all part of the NER system, and it's a state-federal partnership. And there's actually a leading agency within part of the state. And our leading agency is Hawaii Institute of Marine Biology, HIMB, and you can see these partners that's listed here. And so in Heʻeia, you can see the pink border is the ahupuaʻa of Heʻeia. You can see in yellow is the Heʻeia NER boundaries. And we work with a lot of co-management partners. Hiʻilei is here representing Paipai'o Heʻeia. We work very closely with Kako'oiwi, which stewards Heʻeia wetlands. And then of course with the LNR and many of the partners that's listed here. So as many of you guys know, if you've come out to Heʻeia in the last, in the last, actually, many of you guys have come out, many when you were kids, in high school and stuff too. It's changed a lot, right? So, less than 100 years ago, you can see this thriving agroecological system and lots of productivity in all areas of the landscape, as you can see. But, land use change and different changes in how you use different areas of this place led to erosion. So then mangroves came into play in 1922. And as you can see in this 2005 picture, all of this dark area is mangroves. And they are all like 80 to 100 feet tall, you know, and many of those people who grew up on that side call this the Stinky Bridge. So this is the view from over here. And they say, yeah, I'm going to enter this like super stinky tunnel where you can't see anything, right? But then the last 4 years, we've embarked on really, really intensive restoration. And we're happy to say in 2022, we can actually see Hiʻilei Kawelo and Kaleonani, and you can actually feel the breeze running through the area and a lot of the spirit that's back in this place. And this is all done because of Laulima and the dedication of, you know, the kiaʻi of this place. These are some numbers, but really it's, you know, restoring through this like vision of Indigenous resource management. And throughout this, so the NER came in and, you know, one of the things that we really advocate for is reciprocal science. So what does this look like? You know, guided by Kaleonani that was developed here, you know, with many of the partners that are here today. And then we published this collaborative research to inform adaptive management and actual framework for how we try to do research in the NER. And these, you know, when you read about it, it seems very intuitive, but, you know, we want to try to conduct community-driven science that actually thinks about its ramifications on the community and actually co-develop research from the start of the project. You know, what does it mean to be partners right at the onset or even beginning of when the project actually takes shape? Co-produce new knowledge and like think about what actual products look like, and then inform actual policy or results through that sharing. And so one of the things that we've done in the past 4 years is to create place-specific research protocols that actually talk about or create these, this is like a flow chart, and what you can take away from, you know, it's really small, but this feedback loop and stepwise process to prioritize needs of the community. And I kind of want to bring on Hiʻilei. Can you say a few words about like how we co-develop this protocol with you guys? Yeah.
Okay. Hi. I'm giving her funny looks.
Yeah, I know.
She's all serious over here, and I'm gonna like totally make a joke.
Yeah,
But
make a joke.
no. But yeah, you know, this has been a work in progress, and it's kind of culminated as a result of many years of working with researchers and kind of like fine-tuning it to a point where it's mutually beneficial for not just the stewards of the fish pond, the researcher, but also the place. Yeah. So I mean, you know, a researcher will come up with a project idea and pass it through us. That's not exactly what we mean by collaborative creation of something. What we'd like to really see is, you know, that individual has a relationship with the pond and is coming up with ideas while working alongside, you know, the folks at the fish pond. Like that is like the ideal, right? So everybody gets it. But there's a lot of back and forth, you know, from, you know, kind of like this project idea that's curated and co-created between the individual and our organization. It's presented to staff. Staff has opportunity to make comments and suggestions. And then that individual is able to refine their project idea potentially, and then it comes back to us for final approval. But yeah, I think the point of it really is to make sure that there's a relationship, right, between the individual and us, and the individual and the place.
Thanks, Hiʻilei. Yeah, like she said, it's also giving the people of the place that kind of like agency and space to say like, oh no, let's switch directions here, or you know, we're actually really interested in this here, you know, so that's really, really important. And then one of the other things that we've done in the first 4 years of the NER is to identify these priority research topics. So there were many focus groups that people did before the NER was even designated. There was a Science to Management Symposium and all of that. So, we put that all together and came up with some priority research topics that are really important to the people and the restoration and activities in Heʻeia. So what does that look like? How do we even implement it all? These are just some faces of our grad students and collaborators working in these different realms of these topics. And then, as you can see, there's a lot of research in Heʻeia. So these are different symbols and colors that represent different research topics. And I'm not going to go into all of this today, but I wanted to talk a little bit about what the Heʻeia NER as a core does to measure some baseline measurements that the projects that we're going to talk about with the DNA is based on. So we just installed our 4th long-term monitoring sensor network here at Papahanaukuaʻola, and this was in collaboration with USGS. So this is really special because this is actually amongst the NER system network as well. So we actually, this is using the sensors that's used in the national network, and it's hosted online using centralized data management office. And so there's 4 water quality stations, Waima'ama, Reef 9, Ka'o'okele, and then at Papahana. And then we also have a weather station at Lono Circle. And Kaleo later on will talk about what the stations are and what we're measuring. But I wanted to talk a little bit about how we even did this. What does co-development of the sensors look like? And these are pictures of how we co-develop. So, from the locations to the actual building and creation of the materials to the installation, and then using the data and then applying the data. So all of this was done with consultations with our partners. And it's from NOAA standards, maybe it's taken a little longer for us, but we were really mindful about, basically putting something like a giant solar panel in a cultural landscape. And how do we do it so that it's, mindful of the place? So we're really proud that, we installed the system and that it's taking really, really good measurements for us. And after 4 years, we've actually amassed some data so we can say a little bit about what it's looking like. And so these are, I want to say thank you to our technicians, past and present, Gus, Noel, and Claire. And then of course, Kim Folinsky, who's been doing a lot of work in Heʻeia for many, many years now. And we worked very closely to get this data and plot it out. So we have we can say some things about, how the inorganic nutrients are moving throughout the Heʻeia ahupuaʻa, what dry season dynamics of nutrients can be and what nutrient limitation dynamics can be. But happy to answer questions about this at another time. We're not going to go into depth about this today. But what do these folks who are on the ground doing the sensors say? They say nutrients change dramatically across seasons and they can impact primary productivity. So how are we measuring the biology of it, right? So that's where the DNA comes in. And we're using eDNA as an investigative tool in Heʻeia. So we're asking questions like, what lives there? And, we see these species, but is it what we expect in our data? Can we actually see it in our data? Can we detect changes over time? So this is a quiz to the audience. We can't answer this. These 2 species of fish are actually very different species. Who can say which one is which? Or can you shout out, what's the top one?
High.
Yeah.
If you don't
This is a pua'ama size of top. One answer. Ama, very good. And then what's the bottom one? It is kanda. It's an Australian mullet. Handsome one too, huh? That red hair. So, okay, so some of the benefits of using DNA, it's easy to sample in different environments. Like we can go up in a stream that's super shallow. We can actually go even when it's deep, and we can use automated sampling methods that Kaleo is going to go over today. And we can detect species that we can't sometimes see. So when it's super murky, we see these species, they look very similar underwater. And we can do species specific detections. But there's also caveats too, where sometimes you can look at community composition, but it's more relative, and then maybe it's sometimes like only presence absence. You can't get to like how many fish there are. You need local voucher specimens. So sometimes it'll be like, this is a species that's found very closely related to a species found in California. And you're like, that's not it, you know. Longer time to process samples in terms of like all the steps that we have to go through. And then we need laboratory and computing access. So these are some of the things that we think about, you know, and we're going to talk about later about increasing access to eDNA. And then this is a brief overview of eDNA sampling that applies to all the projects that we're talking about today. So basically you collect an environmental sample that has traces of DNA in them or actual DNA of microbial. So it can be water or sediment. And then you concentrate it down somehow onto a filter or material. And then you extract the DNA, you purify it, and then basically amplify means you make like millions of copies of the actual primer that you're interested in, whether it's for fish or plankton or whatever it is. And then we sequence it and identify it. So that's the overall steps that we've applied in all of the projects. So Aka's here today, and I'm presenting some of his work from his master's. But you know, one of the questions that we've asked for a long time is what is in Heʻeia Fish Pond. And this Heʻeia Fish Pond time series is led by Rosie Alegado and the Alegado Lab. And I have the privilege to mentor many of the students in her lab, and they call us the Shiro Lab, as shown here. And so what Aka did is, you know, you can ask him after the talk here, but he used species or group specific primers to look at diatom abundance in the pond and how they were found where, and he found that they were highest in areas of intermediate salinity, so brackish areas, right? That follows along with this, like brackish waters are really good for the fish pond. And then he also looked at and saw these different campaigns of restoration around the fish pond and saw that restoration was linked to a colder and fresher fish pond. So there's some other grad students, Becca Lensing and Huaka Thomas. Huaka's here today too, from the Alegado Lab, and Sarah Tucker from Micropay Lab at HIMB. They've also worked to identify plankton in Heʻeia Fish Pond. Here's Sierra Ratum, she was our summer intern, and she used the flow cam to actually visually look at what plankton is in the fish pond and what types of diatoms are in there. And then you can actually like match what you see visually versus what we see in the DNA. So this is a crazy bar chart, but you see something like, you know, it's mostly diatoms or dinoflagellates, and where are they found in the fish pond. And then, so one of the cool things that we've seen too is that we're actually seeing community shifts. This was a data plotted by Sarah and Becca, and we've seen this community composition of like percent of diatoms that are the smaller single-celled cyclotella to this chain-forming larger diatom Chaetoceros from 2015 to 2017. And we were meeting with Hiʻilei folks and being like, what do you think happened? You know, why do you think there was this like big shift? And we were kind of talking and thinking maybe it's Panikapuka or...
I was gonna quiz you guys.
Oh, sorry. We won't say that. And then what do you guys think? What do you think happened? Oh, Katie, you get pie and a Subway sandwich. Yeah. So we really think, I mean, this is how we piece together stories, you know, like we think Panikapuka happened in December 12th, 2015. And it was this big event where we closed the gap in right by where Kahoʻokele is, Ocean Break. And it changed, you know, and Aka showed this too, and basically changed the geochemistry of the fish pond.
To add.
You want to add in?
No, I mean, just think about it, right? Like I'm a real visual thinker, but you know, prior to Panikapuka, water's flowing in and out of the pond, kind of all over the place, right? As restoration progresses. Then we close the puka on December 12th, 2015. And for all intents and purposes, the entire fish pond wall that's open to the ocean and estuary is closed. So then the fish pond, kind of redistributes the amount of the volume of water that's leaving the pond. It's more evenly distributed. Because prior to this, a lot of it was going in and out through this big puka in the wall. So basically what happened was, as the fish pond is restored and better able to retain water, both saltwater and freshwater, it actually increased the residence time of freshwater that stays in the pond, right? So in essence, making the fish pond a little bit more fresh than in what it had been in the last, 20 years of the organization plus, or even the last 50 years. Which is good for phytoplankton. That's what the slide is showing, right?
Yeah, lots more nutrients and, these bigger chain forming diatoms that, a lot of the fish like to, and contributes to the food web. Yeah.
Is this Sarah's work?
So this is a time series that the Allegado Lab did, and then Sarah and Becca and a lot of people have been kind of working together. But yeah.
Yeah, which is really cool. So one of the earlier slides, you saw that there was a location named Waima'ama, and that's actually a new name that we created. We named that particular second freshwater makaha Waima'ama this summer after building the hale kia'i there. But it really speaks to what we see and talk about co-creation of not just projects and work, but knowledge creation, right? The work that all these guys had shown, and Sarah, in addition, who is that? Yeah, we're a little bit overly matchy today. But her work shows that, ama may potentially be more ono for those chain diatoms, which are actually larger in size, which proliferate at that particular location in the pond, which totally is cool because that's what the name says, right? It's talking about the vai, the freshwater ma'ama. That is kind of like where we see the ama hanging out because they love it there so much. Speculating as to possibly why they love it there so much could be the abundance of those chain diatoms and the proliferation of them specifically there. But ma'ama also means like light. Ma'ama means light. So it's kind of cool.
Yeah.
And we didn't know that we were naming the makaha because of the work that was going on and the research that was being generated, but it just is serendipitous.
Yeah. Chills. Okay, so that's kind of the plankton in the fish pond. And the other thing, and we're leading to some of the specific projects, is we're trying to see what kind of fish are in Heʻeia, not just in the fish pond, but in the stream. But we use 3 different methods because they're all so different. So we have visual method that we do every 6 months that go all the way up to Papahana, where we are like snorkeling. And then in some places we're actually like doing visual surveys. We do cast net, throw net surveys at the Muliwai, and then we actually do eDNA surveys right now bi-monthly all the way up the watershed. So some of the things we're doing, we're looking for oʻopu, right? It's a really culturally significant species, and they're supposed to be distributed across this elevation. So, and then that tells you kind of the health of the watershed as well. So, how many species do we have in Heʻeia? And we can actually see some of them, sometimes like when we're not doing surveys, but can we actually see it through eDNA? So this is a really brief snapshot of what the visual surveys tell us. So red is non-native species. All the streams are all like cichlids and all the aquarium fish. And then the blue is native species. That's usually like aholehole, ama, all the ocean species. But preliminary eDNA surveys has actually shown these 3 types of oʻopu, the akupa, naniha, and nakea, where we see them and we can see it in eDNA. So that's really, really cool. And then when you actually try to evaluate all of our methods together, and it gets to be really complicated, but we see this lots of overlap with the eDNA. So eDNA is the green. And so we continue to do all these methods because continuing to merge the data sets will get us a fuller picture. So there's some species we don't see in some of the methods, but we can put it all together. Okay, so we're gonna get into kind of like chapter, I don't even know, 2 or 3, but we're gonna take you deep into the Heʻeia stream. So you're flying into the stream, and that's right around where Waimaʻaʻma is. And pretend you're this fish, you're just hanging out in Heʻeia stream. And that's actually what it looks like. There's our sond. And let's pretend it's July, right? In the middle of summer. And right after the new moon, you know, it's about brackish, salt water, ole moon, things aren't going well today, back up your hard drive, but you know, it's about 10 PSU. And then, it's getting fuller. Hey, how come it's getting super salty, right? What's going on? And then it's full moon, and you're like, it's getting very salty over here. What's going on? So then if you're this fish, what do? Do you actually move upstream? Do you move downstream? But that's kind of the premise for this next project that we're going to talk about. I'm going to hand it over to Kaleo.
Aloha. Yeah, so that's the big inspiration for our PI-CASC funded project, which is a collaboration between the Heʻeia, between Kua, Paipai o Heʻeia, and Kakoʻoiwi. And essentially the big question that we're trying to understand is how are these stream fish communities going to change, and especially during times of extreme changes that we're seeing. So the main goal of this project is to look at that. So how do we figure out, how do we find what the fish communities are? We give that away a little bit in the environmental DNA. So it's essentially that, organisms living in the water are sloughing off DNA all the time through the mucus, scales, dropping eggs, during reproduction, carcasses. There's a number of ways that DNA gets into the environment. And so that's how we're sampling, is by looking for, not actually sampling the fish directly, but looking for those traces of fish by whatever they've left off in the water. And then using that information with what environmental measurements we've been taking to try to see if there's any kind of co-occurrence, or if there's any kind of, predictions between what's changing in the environment and what's changing within the communities. So there's some objectives that we're using to try to get through this, or to get to this question. Essentially that it starts off with reciprocal collaboration. I am from Ewa. I am from the Moku of Ewa. I've lived in a couple of different places of Ewa. I am not from Koʻolaupoko. I have the great honor and privilege to be able to go over and to traverse into Heʻeia and work there. And so all of that just to say that it's not my wahi. And so I don't know the things that other folks who have spent all of their time and all of their years and generations I would know. So someone like me coming into this space might not have as good informed an idea about where to focus some of the work as some folks who have been spending the time. So in that way, there's ways that we've been trying to communicate about, through our surveys and our different ways of looking at fish, target areas, things like that, and trying to understand what kind of information that we're gathering is going to be useful. And then we're detecting and quantifying fish biodiversity, again, through those different survey methods, and also through gathering water samples for environment DNA, and then looking at our environmental measurements. And ultimately, this whole process of, coming into a space and looking at the environmental tools and then looking at DNA, all of it is meant to feed back into this, what we're calling our investigative biodiversity toolkit, because the whole point for us is how do we use this information to equip and to assist resource managers with how they might be able to use this for their questions that they have for their specific spaces. So even though this is a project that's primarily taking place within Heʻeia, a lot of the ways that we're going about the work could be translated or interpreted in other spaces if those resource managers find that to be of use for themselves. So this is just to give you an idea for the scope of environmental measurements that we have. So we have a number of sites throughout the ahupuaʻa, within the bounds of the NER and also outside. We have sites that we have continuous 15-minute monitoring measurements with our sondes, things like temperature, salinity, depth. So this is continuous. So that means that it's taking these measurements every 15 minutes. And then in addition to those continuous measurements, we also go throughout the ahupuaʻa and do single point measurements consistently the same time of month, every month. And it takes a whole team of people to do this, which is really amazing for me to be part of the team. So there's other water quality parameters that we do on the monthly, which are things like looking at productivity, chlorophyll, nutrients, and also environmental DNA. And what I think is really amazing is being able to see all of this data in a centralized database that you can access. So this is data that is collected at the long-term monitoring sites with the SONS that goes, that's again, is housed online and you can access it. And it's especially useful, again, when we're looking at DNA in the environment, being able to see what was happening in the environment at the time of collection. And so this is just to give you an example of some of the data you could access. So you can compare parameters within sites, or you can compare them between sites. So the two examples I have over here is one of our stream sites. It's within the stream, our oceanic site, which is just off the Kuapa, the fish pond wall. And on the graph on your left is looking at depth. So the blue bars and the fluctuations in blue are measurements taken at the oceanic site, and the green measurements are taken at the stream site. So this is a comparison of a single parameter, depth, between two different sites. And so you can see there's these fluctuations, which if you can't see the time, which probably you can't on the bottom, this occurs over a couple of months. And so you could maybe guess what those fluctuations correspond to in a month. Anyone? Tides, yeah, totally. And it's really cool because you can see that, right? You can actually see the fluctuations in depth. And then on the flip side, we see changes in salinity, which is perhaps not unexpected for, again, the blue line is oceanic. So you're seeing that there's little minute fluctuations in salinity, but essentially, you know, it's relatively similar. But there's major fluctuations in the stream. Like the magnitude of fluctuation within the stream is like, this is not a freshwater only site, which is really something you might want to know if you're looking at mobile organisms such as fish that can sort of have a choice about where they are in the water. But again, this is just long-term monitoring. This is just data that we are able to see, observe with our instrumentation. And one of the reasons why we're interested in something like this and being able to have this kind of information accessible is that we are trying to make sense of impending changes and actually changes that we're starting to observe now. And that brings us into the climate aspect of this, which are the extreme or the king tides. And so in Hawaii, our extreme tides happen pretty much every 3 months. And so in the summertime, in Ho'oilo summertime, it happens during the, in the wintertime. Oh my gosh. That's okay. It is because we are in Ho'oilo, but it is not May through July. That should be November through January. So the new moon is when the extreme tides occur in Ho'oilo, which is actually our wet season. So that would be now. And so essentially what happens is, you know, the tide changes throughout the month, but there's these periods of time where the moon is at its closest on its axis rotation around the earth. So it has an extreme pull than it normally would. And when that combined with the proximity or the relationship with the sun happens, basically you're getting this huge tug on the water that's beyond what we would normally see. So in the winter time, which is now not May, July, but November, January, it's very confusing, I know. The Hawaiian is correct, the English is not correct. So during the times like now, this occurs during the new moon, but in the summertime, it actually occurs during the full moon. And you can start to see those things when you look at our long-term monitoring data. So I just wanted to show you some of the examples that we have of what can occur when the predicted king tide actually translates into extreme flooding in some of our nearshore coastal environments. So such as flooding of the fish pond wall, which is not an intended thing, right? It's but it can occur. And then there's also things like flooding at the stream mouth, and that area normally, I mean, it is a stream, but the area that's in the foreground of that photo should actually be dry land. So these are just some extreme examples that have happened again during our king tides. And we are trying to understand by measuring during these king tides, how might these serve as proxies for what we could expect during sea level rise. And that's what that image on the far right is. Essentially what could be flooding within the ahupuaʻa in future predicted flooding scenarios. So basically all of that area that's a little bit upland might potentially be covered with water. We want to know that. Okay. Great. And so how, though, do we then go about detecting how far upstream saltwater intrudes or how far upstream water will move? And so that's how that's what we've been able to do using our hobo loggers. So with conductivity, which is a proxy for salinity, and the water levels. And so these are three of the sites that we focused in on: the lower wetland area, which is in this diagram, the more upland of the three, this middle lower stream area, and then Waimaʻama, which is sort of the lower of the stream sites. And so basically going from left to right is going from upland down. But actually all of this occurs within a really small area. I mean, I think I did a calculation on Google Maps. It was like 165 meters. Like it's not a very large area, and yet there's major occurrences. So for example, from some of the data we've been able to collect from keeping our hobos out for a month, we can see that, again, lower wetland in the green, you can barely register it on the salinity plot. That's because it's entirely freshwater. The fluctuations are very small. And yet in our middle stream, where we have sensors at the top in red and at the bottom in deep, you can see that it actually goes to almost entirely saltwater in this middle stream area at the surface, and yet not, I'm sorry, in the deep, and yet not at the surface. So there's a big change happening over there. And then Waimaʻama, which is again where our sonde is, it also shows big fluctuations in salinity. So again, within this 165 meter area, there's like this big change. And again, we're looking at fish who can move through these areas. We want to know about access, things like that. So this is sort of helping us to sort of understand what things to look for within an area based on environmental measurements. And then we also have targeted environmental DNA sampling, which is essentially when we go out and do our collections. It's based around when we do the other visual surveys, which makes sense because we already have visual surveys going on. We want to use that sort of as a way of checking that our methods are working. So we would hope that we could detect in DNA what we're visually seeing. But if you are not detecting it in DNA, that is also something interesting to know. And then we have specific king tide sampling, which occurs again during our November, January, May, July windows. And then we also have the in-between ones, which is our baseline. Because again, how can you say it's an extreme if you don't know what's happening in normal? But I think thinking about what normal looks like under changing conditions has been a really interesting thought exercise. And that ultimately most of this environmental DNA sampling occurs with hand sampling, but we also have this neat little machine, Teledyne ISCO, which does automated sampling. You can tell it when to sample, you can tell it how often to sample, you can tell it a volume, it does all the things. I love it. It does all these things that you then as a human observer do not have to go out 24 hours, especially if you have small keiki to go into. So it's really cool. Our team nicknames it R2, but I think it's more of an R5, R6 unit because it's got the square top. It's not rounded like R2. Also, it doesn't have wheels. That would be so cool if it had wheels though. If anyone out there engineering, yes, okay, cool. So we take all these water samples, whether they've been hand collected, whether they've been collected with the ISCO, and we bring them back to the lab. We do all this water filtering, tons and tons and tons and tons of water filtering, which when I was an intern, I said I'd never do again. So what I've learned is never say never. But really cool to actually be able to look at the filters and see the actual differences, because in these bottles, it just looks like water. And then you look at it on the filter, you're like, wow, there's stuff in there. So the next phase of that is figuring out what stuff is in there, right? That would be the lab side, the extractions and the quantifications and all of that. So again, this is just to show you the overview of what our sampling has looked like. It's important to get our baseline and then sort of using that as a comparison for our extremes. And then this image that we have on the right is showing you over a 24-hour sampling going from the highest tide down all the way back up to the highest tide. That's what the water can look like in a 24-hour period at one site. And this is our very dynamic site. And you can see that the water is more clear, less dirty during certain times of the tide. Something we'd want to know about. And then ultimately it goes back and forth between working on environmental data and then working on fish data. And so we have a multitude of samples that we're getting through. We've had visual surveys, again, that we've used for comparison. It's good for checking our methods. We hope to find in DNA what we are finding visually. And then also because we also have some of these potential hands-on exercises or observations like their CASNet, we can do things like fill in holes to our DNA references by sampling. And so again, I mean, one of the cool things I wanted to point out is this stripy fish, which is actually nicknamed a stripy, which was a surprise to find because apparently they're actually nocturnal, and yet it was upstream. They're nocturnal reef fish. So that was a surprising find that we were able to see because of CASNet, that if we had only seen with DNA, we might question the accuracy of. And then to try to wrap it, again, all of this process of trying to figure out what methods work, what are some of the challenges for these methods, challenging being, you know, I said we lugged lots of water around, but if you were a resource manager and I told you had to filter water, what might that look like for you? Changes that you can see, and then things like their environmental events that we can sort of predict. We can predict that king tides are going to happen, but sometimes the reality of that king tide is not what we expected. There's also flooding from upstream. How do you sort through all of that information? And ultimately, what does it look like for accessibility for resource managers? Because that was a lot in a short period. I know, because I talk really fast. But what does that translate to for actual use for resource managers who are in a space?
Thanks. So I'm getting the eye from Rachel, so we gotta hurry it up. So I'm just gonna slide through these slides real fast because I really want to talk about our collaboration with Brenda and KUA. But this is another example of a project that we're doing that's actually Sea Grant funded. And our students, Sheldon and Lunny, work with us on this project. But we work with Hiʻilei. This question really came from you guys, right? About, why you're interested in this fish. I don't know, maybe you can say a
Real
word.
fast.
Yeah, real fast.
Real fast, it's pretty obvious. We're a fish pond. And it's 800 years old, and our fish ponds grew fish for consumption. And this is like the keystone fish pond fish, right? It's actually what I grew up eating. And if you do if you do any kind of historical research, you know that every single traditional Hawaiian fish pond boasts that it has the biggest, fattest, sweetest mullet, right? And this is the species we're talking about. So in our work to restore the fish pond, we're trying to restore habitat and ecosystems to be able to cultivate this fish. And because there's been so much phytoplankton work, so this is the transition into this particular project through Sea Grant, is we're trying to understand what they're eating. Like, what are they, if maybe they're selecting for a specific type of phytoplankton, who knows? Maybe they're just opportunistic, but if they are selecting, is it possible that we could somewhat mimic the environment that's required to allow for the proliferation of that specific diatom versus other species of phytoplankton? So we love to learn about fish. For so many years, we've learned, we've been studying water quality, water chemistry, and it's still relevant. It's still super important, but it's for us as kia'i of the lokoi'a, this is what we're asking for. This is what we would like to learn a little bit more about, so that if there's a way to create an ideal environment within the fish pond, if we need a little bit more fresh water, or a lot more fresh water, or we're good right now, like we can have those conversations with people like Board of Water Supply or the Water Commission. If we need more fresh water, maybe they can help make it happen.
And what's like really special is like, we're like, okay, how are we going to catch more fish when we actually want to preserve this fish? So then we kind of came up with this plan to basically harvest the guts from the fish that Hiʻilei them are already collecting.
Right. I think a lot of times, right, when you're given a research proposal, it's like, oh, we need you to catch 20 mullet in this size class per month. And that wasn't going to work for us, because it's such a coveted species for us in the fish pond. We want to see more and more and more of them abound in the waters of the pond. That for us, what was doable was we're just extracting stomachs and gonads and other bits of information from the fish that we're already catching. And because we limit ourselves to one per employee per month, and we don't actually always get that. It's kind of a special thing for us to be able to eat a mullet from the fish pond.
So this is some of the work that Sheldon and Lenny and we're going to be working with students from the OPE program as well. But we're gathering, sediment and trying to partition out where the food that mullet like to eat are in the fish pond and match it up. And then I'm going to skip this, but there's super awesome Sheldon at Resiliency. If you were able to catch him, if you didn't catch him, I think it's online. So you can go there and learn all about Anaiholo. And this is also another collaboration we're doing with Brenda at KUA, because stay tuned, because we're going to do some cool events in January and beyond about trying to mimic the migrating mullet throughout Oahu. Okay, so I'm going to turn it over to Kaleo and Brenda to finish us off about what we did in terms of the community eDNA workshop.
Awesome. Yeah, so one of the steps for us trying to figure out what reciprocal collaboration looks like and what information we need for a toolkit, if we were going to, again, be reciprocal about that, is how do we communicate this information about what we're learning and what we're learning in problem solving and doing the work with people who might actually make use of it, either for trying to implement it themselves or because they are constantly being approached by researchers who want to get into this not new, but new application of this tool of looking at DNA in water and what it means. So we got together and tried to figure out what at least the first step. And the first step we had was we had so many ideas, I think, right? For in the beginning, I think we dreamed about oh, maybe we could do this multi-space, someone on different islands, and we host this simultaneously simulcast event. We get together, we nerd out about fish and DNA, and that was maybe not feasible.
Yeah, maybe a little too big at first. No, but I think so what you're seeing on the right side is what ended up being a small workshop at Waikalua Loko on Oahu, but through the existing network of the Hui Malama Lokoi, making that an opportunity for other kia'i to learn about this tool. And what Shimi said way at the beginning of the hour, to start seeding in people's minds, what is the agency that they can have to apply this tool in their places. Just start to learn about it, talk with other people about what kinds of questions it can answer. Really understanding the tool itself, I think, helps folks like Hiʻilei and other managers to know how it can answer the questions that they have. So I think this work will continue to grow and, just try to reach more fish pond sites so that they can learn about this ongoing eDNA work in Heʻeia.
To just kind of fast forward through the preview of some of the things that we did. We had one activity at the very beginning, because this question comes up, not surprisingly, a lot when you say eDNA at someone, which is, what even is that? Is that DNA that exists in the computer that you email someone? That's why it says electronic DNA. No, so it's it's starting at the foundation of what even eDNA is. And so we actually have the activity in the back. You wanted to check that out afterwards. As a tool, again, to demonstrate, I guess, more simply or more basically, what information can you gather and about the process of doing the work. And then we went through an activity about actually collecting the water, and all of the various questions, just to name a few of things that people will ask, have asked, are going to keep asking. When do you do it? Where do you do it? How do you do it? How much water do you have to get through? What is it going to cost us to do this? If we're going to try to implement this as a monitoring, are we going to be able to afford to do this at all of the sites that we're actually interested in every month? Or is every other month? And these are all questions that, of course, research is meant to address anyway, right? These are things that we're all thinking about all the time. How often, how much am I doing enough to do this thing? But this is more on like, I would say a little bit of a practical scale of like, no, really, how much is it going to cost? And what resources am I going to have to divert to allow for this to keep happening? Am I going to have to bring in outsiders to do it? Am I going to be able to do some of it myself and ship some of it off? And that's some of the questions that we're trying to get into answering, or at least addressing or talking about a little bit in our toolkit.
Yeah, and I think this aspect is important because for folks working at these kinds of places, like again, a lot of times they're approached by people that have questions and have a sampling method in mind and kind of know the scale and how often and where they're going to sample. But for folks to actually see how it's done and think about like how often and where might we want to do it, and to just have the open space to ask these questions without an agenda about like what the project is going to be, I think is a really cool space that was created by this.
Yeah. And then just one of the products that was generated from this is again being able to take a step back for us and think about what kinds of questions could we help with guiding. So these are not questions for us to answer within our project. These are some questions that we get asked often enough that it seems like this would be a good place to start. So the first question is like, what questions do you even have about the place, about your space, your place? What are some generic or basic or whatever? What do you want to know? And then other things like, what are things that you're already monitoring, or what are things that you want to be getting into monitoring? You know this, more about this than that. You want to know about that. That's a great question. Or what kinds of things are you already doing? Some folks are already doing their own monitoring, water quality, nutrients, things like that. So then, what are you doing? And then that kind of gives you an idea for building off of that. So these are just general questions that we've been putting into our worksheet, which again is a way to help folks really narrow in on, because we want to know everything. Obviously, what questions do you have could take up more than this little box could possibly hold. But this is a good jumping off point again for resource managers to be able to I guess kind of determine on their own, this is the direction we're interested in heading, because these are the things that are priorities for us, which as a researcher not from that side of the island, I couldn't possibly determine for someone else. And then also trying to include things like case studies, things that other research groups have done, because Heʻeia is not the only folks working on environmental DNA. A lot of people are doing environmental DNA for fish. The state's doing it, for example. There's other labs within UH doing it, things like that of like, these are projects, this is the questions people have, this are some examples of access. And then definitely recommendations and best practices, things about how is your data then going to be stored, the data that you are collecting physically, but also the stories that you are collecting by writing down observations, and how is that going to be shared? How are you going to have access to permissions for things if you're going to be presenting as a researcher out of the projects to a wider audience? Things like that, logistic. I mean, we have big dreams for what's going to happen with it. So hopefully we get to see some of that come together for that.
I think that's it.
Yeah.
We can talk through that. We want to thank all the people who we work with that, do the work every day that are here today presenting with us. But thank you guys.
Thank you very much. Yeah, excellent work. We have some questions online, Rachel.
I should.
Yes, we can start with questions in the room for 2 minutes. Or we can rotate, but let's start with the question in the room. We got one. You're sampling for DNA from Heʻeia. How many do you have? And the fish move around. How many do you need to cover? I mean, for example, we want one every foot, for example, a fish is like being every quarter mile. But I'm just curious.
Yeah. So for the folks online, the question is essentially about, you're working in moving water system. How do you decide, or how do you figure out how many different sites you need so that you're sampling in a way that makes sense? Because you're right, like you don't want to sample, every foot because you're cross sampling the same organism. But also, we're dealing with a stream, so the water is flowing in one direction. And for us, it's all about timing with the tides. You don't want to be sampling, or if you're gonna embark on this journey. You want to sample consistently. You want to sample when it's low tide, for example, which is what we do. So we know that the majority of the water is actually moving out downstream. And for us, it's been a matter of working backwards, going upstream, so that whatever we're sampling at the top is not what we're then sampling at the bottom, theoretically, again, because the water is moving. Things like that. So we currently have 12 sites, including a double site where we're sampling at the surface and at depth. And the double site area is our dynamic site where we're seeing a lot of fluctuation of salinity. And that one makes sense because, you know, as we've shown, like even at the surface versus a few feet underwater is a different environment. So things like that. And those are all important considerations, definitely.
So DNA doesn't last for a very long time in the water too. So, you know, us seeing those like opu in different areas was really like awesome, because that means, you know, we're not seeing Nakea DNA at the stream mouth, basically, right? Like we know that it's persisting there. And then by the time it gets to the wetland, we're not seeing Nakea DNA. So, you know, those are all things that we're figuring out, like DNA decay rate, as well as, I mean, we're not doing the experiments, but that all goes into your question.
Thank you. We're at 1 o'clock. I want to make a couple of announcements. I know folks start logging off here pretty soon. And then maybe you would mind sitting down and answer a few more questions. Unless you got to run. Okay. Just to let folks know, we're going to take a break from our PI-CASC seminar series in January, but we are doing our Hawaii Climate Week and Climate Adaptation Science Summit that second week of January. So hopefully you're starting to see the announcements for that, and we'll register and join us at those events. We'll be featuring a lot of great science and partners through that event. We'll be back, and that's January 9th through 11th at the Min Conference Center at East West Center here. So just a short walk from here, and we'll be broadcasting that online as well for folks that aren't here on Oahu and can't join us. So we'll resume our PI-CASC seminars on Tuesday, February 7th, again at noon, Hawaii time, where Dr. Katie Kamelamela will talk about her work on investigating the cultural dimensions of Indigenous local knowledge responses to changing climate conditions. So please look for sign-up information for that to come in January. Check for that online. And as you're logging off Zoom, please do take a couple minutes to complete the quick survey. And we thank you all again for joining us today online and in person, and hope you'll come see us again. And mahalo again. Big mahalo to our speakers for joining us today.
We'll pass along the questions from online. Yeah, guys.
Yeah, were there any other remaining questions in the room before folks take off? Yeah. Look at mine.
So stream site, it's really salty. And then Shimi posed this question like, if you're a fish, what do? Are they moving further upstream? What happens to the fish on a home today downstream? So they just like staying put and they're like, it's really salty. And I'm just gonna stay here until it's less salty.
So we don't know yet. Because we've collected a whole bunch of samples and we're in the extraction stage, and we haven't actually put together. So like a lot of the environmental data we have, but we don't have the fish and, you know, the composition data. But yeah, so we have like low tide sampling data, but then we also have like at high tide, we've like launched like 4 teams and sample at the exact same time too, you know, to see if the community there is the same as the community upstream. So stay tuned.
All right. Thank you so much. Oh, yeah.
I just saw a couple screen. It was a really interesting question about data sovereignty.
Oh, yeah.
You guys like discuss like protocols for that.
Oh, yeah. Saw that too. Let me relay it from Rachel's.
Yeah.
Okay, there was a lot of compliments for the great presentation. Yeah, again, this one talks about great stuff presented here. If you had any discussions around data sovereignty, what are the collective benefits and who has control, authority, and responsibility for the data, the ethics of its use, how, why it's being collected. Stop there.
Yeah, so in that research protocol that we described, there are things that people have to actually initial. Trying to get at that question of like data sovereignty and who has the right to actually say, yes, that can either be presented or that can be used. And we try to put into place those kind of sanctions, so that people, I mean, basically for us, like all of this data belongs to Heʻeia. So, and then, we try to leave out some of the, just because we're protecting like specific names and specific, partners and things that like we're working with, because those are things that like maybe should not be shared, without specific permission. But I want to give you guys space to answer that question.
Yeah, I mean, it's definitely important. For me at least, it's a constant kind of thought, because I mean, it can be even as simple as someone from the area sharing about observations that they've made over time. Is that something that I should then go out and share? This uncle told me to say, or this auntie told me to say. For me, it's like it's easy enough for me to think about those things, but those are questions that we're thinking about even for the toolkit when we think about, so we have this worksheet that's for the resource managers to think about their own questions, but then we're also tying into like the Kaleonani Hurley and taking some other direction from there. What are things that you as a manager might want to explicitly say about having protections over presentations, especially because everything is online and people can screenshot, you can record so much more easier than they could before we had this online universe. And so I think for things like that, it's a matter of going back to the reciprocity part of it, which is, this is a really cool piece of information that I learned from the sampling, but is it up to me to decide as the researcher how to present that, or should I actually be talking to the people managing the space where I got this information from the first place, because I wouldn't have been able to do that without them anyway, right? And then even things like, yeah, we want to share that out, but here's the way that we want to share out. Things like that, and it's essentially asking for permission all the time. It's a constant process of figuring that out. Because ultimately it's about the question of, for the data sovereignty, it's for whom does this data serve? Right, that's the goal I think to keep in mind.
So you guys have like the infrastructure to sample, conduct research in the community. Like, what direction do you see they're going into? Or like, where would you like to see them go?
So, we're not really, we can just answer in the room. We don't have to repeat the question.
You're okay, yeah,
Okay,
we
okay.
most people log off.
Okay, cool. What direction is the NER going into? I think, in our management plan and where we're at, so the landscape has changed so much, it was really focused on like removal of mangroves and like, how do we measure what's happening during that removal? And then now it's like going into this different stage. So constantly checking in with our partners about what their research priorities are. And then also, like, where do they want to go in terms of like, I think we're kind of established that baseline of like, what's there, who's there, like maybe doing more. I think that was the future fund stuff that I kind of skipped over, but it's like, what are the organisms doing? Kind of like, metabolic and functional questions that we're going to be trying to get at in terms of research. And then even then we've got to start thinking about, like, I think we're with these grants, we can kind of move not out of the area, but like, how do we work together with other communities to be like, okay, people are at different stages of restoration. Like, how do we, work together to learn from each other? So I think that's kind of the launching off point where we're at.
Great, thank you. Well, unless you have anything else you want to add to that, we'll stop here and more sandwiches.
Yeah.
Okay, so pretty much pie, and I don't know if you need to run, but stick around and
Yeah, I'm definitely having pie.
thank you again so much.